Okay, so we are going to talk about the molecule DNA, deoxyribonucleic acid, a very famous molecule. Okay, so here on the left we have this this structure. This is called a double helix. Okay, a helix is a spiral, and there's two molecules which spiral around each other here. So each of those black strands is a separate molecule. The two strands are held together by hydrogen bonds. Okay, now once again, H bonds are not covalent bonds. They're weak intermolecular forces. They're the strongest of the weak intermolecular forces, but they're a lot weaker than covalent bonds. Whereas all of the atoms within one strand are covalently bonded. So that means then that the two strands can be separated quite easily, although to break a strand is difficult, but they can, and that's really important that the two strands can separate. So let's have a look at the structure of each strand. Okay, so I haven't drawn it as a helix in this picture here because that's too difficult. I've drawn it just as a straight line. Now what we have is we have a backbone here, which is sugar. That's a sugar. And it's a sugar called ribose, well, deoxyribose. And then we have a phosphate group in red. And so you have an alternating sugar, phosphate, sugar, phosphate. Now sticking to each sugar, and it's we have, and that actually that's carbon atom number 1 there. We have a base, and there are 4 bases. Okay, so they're in green. So you can see you've got A there, T, G, and so on. Yeah, right. We'll say what the bases are called afterwards. It's called adenine, thymine, guanine, and cytosine. Let's have a look a little bit more detail at the strands first of all. So you may have noticed that this strand is running one way, and this strand is running the other way. So these strands are called anti-parallel. Anti-parallel. And that's also pretty important. Right, let's have a look at the building blocks of DNA. So DNA is a polymer, and you'll probably see that the repeating unit is, well, I'm going to draw it there in purple. That is the repeating unit. So you've got phosphate sugar and a base, and that is called a nucleotide. Okay, so let's have a little bit more detail on that. So again, you don't need to know, but it's important. It's probably helpful. Right, I'm going to draw. Right, this is deoxyribose. Right, ribose has the formula C5H10O5. So you can see it's very like the sugar, like glucose, which is C6H12O6. It's carbon and water, carbohydrate. Same with ribose. Deoxyribose is actually missing one of those oxygens, so it's only got 4 oxygens there. Okay. Now, right. So on deoxyribose, you have an OH here. Again, it's a kind of a flattish structure like glucose is. You could show a bit of depth to it if you wanted to. You have OH there. Now on ribose, there's an OH on this carbon as well, but there isn't in deoxyribose, it's missing. And we have an OH there. And this is, so how many carbons we've got? I'll show you, we've got 1, 2, 3, 4, and 5. That's where the carbons are. Get rid of that. And you also have an OH attached to this carbon here. So I'll just put CH2 to show there's a carbon there, and you've got an OH. Okay, so how does that bond to the phosphate? I'll show that in a minute, but let's see how it bonds to the bases. Now the bases, I'll show you the structure of the bases later, but the bases all have an NH2 group here. And what happens is you lose water. So you're going to lose that molecule of water. And it will stick on. So it's a condensation reaction. So I just get rid of that. So you'll end up with an NH and then the rest of the base there. Okay, I'll just put base. Now on the nucleotide, the building block, you have a phosphate group. So attached here, and a phosphate is like this. Phosphorus forms 5 bonds. So you have OH, OH, and this is an O minus here. Right, again, you get another condensation reaction. So you're going to lose water here, and it's going to stick onto that. So let's draw that. Okay, and that's called a phosphodiester linkage there. So, right, that is one nucleotide. Right, just to show you how a nucleotide, how they can polymerize together. Then, so if I copy that, and then I'm going to paste it down there somewhere. Okay, make it a bit smaller. Right. So I'm going to move that up here, there, and now, right, all I have to do now is remove water here, so you can see you lose H2O, and you've got a dinucleotide. So there's 2 nucleotides stuck together, and that's how the whole thing is built up. So there's a lot of condensation reactions going on there. I'll just show you the way the carbon atoms in the ribose are numbered. So that's carbon atom number 1, number 2, number 3, number 4, and number 5. Okay, and that's fairly important. You might see written down, this is called, so this end here, which I'll draw here, this is called the 5' end, because the last bit is a carbon atom number 5 on the ribose. Whereas if you look at the other strand, well, that carbon there is carbon atom number 3. So that's called the 3' end. So this strand is running 3' to 5', and the other one is running 5' to 3'. So they're anti-parallel strands. Okay, so the nucleotide then, it is important to remember, is the building block. The nucleotide is the building block of DNA. And DNA is a polymer. Unlike a polymer like starch, where you only have one monomer, of course, there's 4 different monomers here, because you can attach 4 different bases. You can have G, guanine, cytosine, thymine, or adenine on them. Okay, so now let's have a look about how those bases pair. Now we can see here in this diagram that adenine always base pairs to thymine, and guanine always base pairs to cytosine. And that's really important in the way that DNA is replicated and how it stores information as well. So let's have a look now at how the base pairing works. So we've got them down here. Now don't worry too much, you'll obviously never be asked to draw these, but you'll probably see them in textbooks, and it's just important to understand what's going on here. So if you look so let's have a look at guanine. You can see this is guanine then. You've got, well, it's attached to the sugar by that nitrogen atom there. And all of these ones I'm drawing in red, these are all carbon atoms, okay? You don't, whenever it's a carbon, you don't write down what the atom is. And also, each carbon atom forms 3 bonds, and so some of them will have hydrogens. So for example, this one here is only forming 3 bonds, so it will have a hydrogen attached to that carbon, but you don't draw it on, okay? But it is there. This carbon here, that will have no hydrogens attached, because that's forming 4 bonds already, and so you get the idea. This carbon, yeah, that one's also wouldn't have any hydrogens attached. This is the one I've just rubbed out by mistake, because that's forming 2 bonds to the oxygen and then 1 to the nitrogen, 1 to another carbon. Right, these dotted lines here are hydrogen bonds. Okay, and if you do chemistry, you will know that hydrogen bonds form between a hydrogen which is attached to nitrogen or oxygen. So that hydrogen is attached to nitrogen there, and they form an interaction with a pair of electrons on another oxygen, so that there's a pair of electrons on that oxygen there. Now, if you do chemistry, you probably recognize why they can form hydrogen bonds there. Now, guanine and cytosine form 3 hydrogen bonds between each other. And if you try to pair guanine up with adenine here, it just wouldn't work. Okay, all the atoms would be in the wrong place to form the hydrogen bond, so it won't pair up with it. Likewise, it won't pair up with thymine either. So guanine can only base pair to C. A can only base pair to thymine. Okay, so GC, they form 3 hydrogen bonds, and you only form 2 hydrogen bonds between A and T. So that means bits of DNA which have got lots of very rich in guanine and cytosine, they're a bit harder to separate the strands than the ones where you've got lots of A and T. Okay, now we need to talk about why this sequence of bases is important. Okay, well, you've got, I forget the exact number, but there are certainly millions and millions and millions of base pairs in the DNA of a single human being. Okay, and that encodes all of the genetic information that makes you what you are. Now, a lot of DNA codes for the sequence of amino acids in a protein. Remember, there are 20 naturally occurring amino acids, okay, and the sequence of those amino acids, they determine the 3-dimensional shape of a protein, which determines the job it does. So the sequence of amino acids is absolutely essential. Now, how is it encoded in DNA? Because DNA, we've only got 4 bases. We've got G, C, T, A. So people realize that if you've got to code for 20, the minimum you can have then is you have three nucleotides code for 1 amino acid. Okay, and we'll cover that later. But so if you had a section of, let's say, 30 bases, that would represent the sequence of 10 amino acids in a protein. And as I said, proteins are usually around 500 amino acids long, even longer. So there's a lot of bases to code for 1 protein. Interestingly, though, it's only about, I think it's about 2% of the human genome codes for protein. Only 2% of your DNA codes for that. The other 98%, people have referred to it as junk DNA, thinking it didn't do anything, but now people are beginning to realize that it does have some function, although not entirely sure what that function is. Okay. Let's see how DNA replicates then. Okay, so there we've got a strand of DNA, a double-stranded DNA, and before it can replicate, the strand has to unzip. Okay, so you have to go from a double strand like this, and it will start to unzip to expose these bases here. Okay, so the hydrogen bonds have to break, and there's an enzyme involved in doing that, and it is of a class of enzymes called helicase. Okay, so it's an enzyme, it's a protein that binds to the DNA molecule and causes the hydrogen bonds to break, and it can work its way along the DNA, breaking the hydrogen bonds as it goes and unzipping the DNA. So that has to happen. Okay, so let's take that as read that that's what does happen. And we've got this strand here. So we're going to unzip this strand. So I'm going to probably just rub out those hydrogen bonds there. And I'm going to unzip it. And so I'm going to remove this one over here. Oh, that's a bit annoying because it hasn't taken this the other bit with it. Let's get that so it takes it all. Oh, it's got taken all the bits now. That's good. Right. So in the cell, then you've got this DNA which has been unzipped and the hydrogen bonds have been broken. And in there, you're going to have a mixture of nucleotides. Now, if you remember, the nucleotides are the building blocks. So you have basically phosphate, sugar, base. So I'm going to just draw that like that. So you can have that's the nucleotide with T. That's in the one with G and C and so on. So you'll have, and the other one, of course, would be A. So you've got loads of those in the nucleus during the DNA is being replicated. So what will happen here is you will get a C nucleotide will hydrogen bond to that. The C will hydrogen bond to the guanine there. This one, you're going to get a G, which will hydrogen bond there. This one will be another C. This one will be an A because it's T opposite, okay, and so on. And then what happens is an enzyme called DNA polymerase, making a polymer, is going to come along and knit all those nucleotides together. So what you're going to end up there with is a strand, okay, which is, and that's going to be T, T, C, G, T, A, C. And I'm sure you hasn't escaped your attention that that is exactly the same base sequence as the original strand that was opposite it. And then what happens with this strand is, with this strand here, is you form a complementary strand. It's like a mirror image, if you like, of it, opposite it. So I draw that there. That is going to, G is going to go there. That'd be C, G, T, A, G, C, A, T, and G. And you've got two complementary strands. And of course, this strand here is exactly the same sequence as that strand there. So that's how DNA replicates. So this base pairing is absolutely essential to replication of the molecule, and also encoding for proteins, encoding the order of the amino acids in a protein. So all the functions of DNA are really explained by its structure.